What's up, everybody? Welcome back. Today we will be discussing the cell cycle and the 5 stages within it. The first stage, G1, the second stage, S, third stage, G2, fourth stage, mitosis, and fifth stage, cytokinesis. They are separated into 2 groups known as M phase and interphase. Let's talk about interphase. Interphase is what is known to prepare the cell for division. There are 3 stages within the interphase. There is G1, where everything but the chromosomes are duplicated. There is the S phase, where DNA is replicated and the chromosomes will be duplicated. And then there's the G2 phase, where the cells grow and prepare for mitosis. There are 2 checkpoints within the interphase. The first checkpoint is right before the end of the G1 phase. It checks for nutrients to see if the cell is a good size and to see if the DNA is not damaged and to make sure the growth factors are good. The second checkpoint is a little bit before the end of the G2 phase, which is the G2 checkpoint, and it checks to see if the chromosomes are replicated and to make sure the DNA is not damaged. Let's talk about the M phase. The M phase consists of 2 stages, mitosis and cytokinesis. Let's talk about mitosis. Mitosis is a process in which the cell will divide itself into 2 daughter cells. The first phase of mitosis is known as prophase. In prophase, the chromosomes are condensed and the spindle forms from the centrioles start forming, and the nuclear envelope, is dissolving. The next stage, which is prometaphase, the sister chromatids are beginning to attach to the spindle by the kinetochore, and the chromatids are moving towards the middle of the cell. In the next phase, which is metaphase, the chromosomes begin to align at the center. And in the next phase, which is anaphase, the chromatids are separated and the chromosomes move to the opposite spindle poles, and the spindle poles move further apart from each other. And in the last phase, which is telophase, the chromosomes cluster at the opposite spindle poles, the chromosomes become dispersed, and the nuclear envelope assembles and the organelles reform. And how there was a checkpoint in the interphase, there is also a checkpoint within the M phase, and this checkpoint, which is known as the M checkpoint, occurs near the end of the metaphase stage in mitosis. It is known as the spindle checkpoint because it helps determine if all the sister chromatids are attached to the right spindle microtubules. Let's talk about cytokinesis, which is also part of the M phase. Cytokinesis overall is the separation of the cell into 2 daughter cells. Within meiosis, there is the phases of meiosis 1 and meiosis 2. Mitosis, on the other hand, only had 1 phase and it didn't have another phase, just like how this one has a second phase where it's meiosis 1 and meiosis 2. Now in meiosis 1, we have prophase 1, where the chromosomes pair up and they begin to exchange. So in this process, this is called crossing over, when they're exchanging fragments of each one. Then it go ahead and moves on to the next phase, which is the metaphase. And in this one, the new pairs, they line up at the metaphase plate, which then leads us to anaphase 1, where the homologues begin to separate to each side of the cell, and the sister chromatids, they stay together. And in telophase, the newly formed cells, which are haploids, and there's 2, and each chromosome has 2 non-identical sister chromatids. Then we go ahead and move on to the phases of meiosis 2, and in this one we have prophase 2, which we start off with the ending of meiosis 1. So we're starting with cells, which are the haploid cells that were made, and the chromosomes become condensed. Then we move on to the next one, which is metaphase 2, and the chromosomes line up to the metaphase plate. Then we move on to anaphase 2, where the sister chromatids will then begin to separate to each opposite end of the cell. And then we have telophase 2, which the newly forming gametes are haploids, and each chromosome has just made 1 chromatid. So by the end of the whole entire process, you'll have 4 granddaughter cells instead of 2 daughter cells.